Thank you very much, Jing, and to the organizers for inviting me to speak here today. So my name is Chris Nam. I am a postdoc with Dr. Jing Yan at Yale. And today I'll be telling you about our ongoing work on understanding how cell-cell and cell-substrate adhesion jointly dictate bacterial biofilm morphogenesis. So in the Yan lab, we study bacterial biofilms. And biofilms are surface-attached matrix-encased bacterial communities. So here is the classical picture of the biofilm life cycle. So a planktonic bacterial cell will attach to a surface, transition to a sessile state, and start proliferating on the surface while producing a sticky extracellular matrix, which is shown here in yellow. And so the cells will grow and divide and grow and divide and grow and divide to form these large structures, these large communities, until a certain time point when, for one of many possible reasons, the cells will decide en masse to disperse from the biofilm and swim away and potentially colonize some other niche. And so the cycle continues. The first thing to know about biofilms is that they are ubiquitous in nature. So biofilms are actually thought to be the predominant lifestyle of bacteria in nature, and they can form in basically any environmental niche, as shown here, including within our bodies. So, and where they can do a lot of damage because biofilm-based bacterial infections are notoriously difficult to treat using antibiotics. So understanding how biofilms form and develop is actually crucial to resolving a vast range of problems across many different domains. Now, from a more basic perspective, biofilms are also very interesting physical systems, and a lot of work has been done trying to understand how biofilms develop their biofilm morphogenesis. And so Jing Yan, my PI, when he was a postdoc, he developed, I believe, the first imaging platform for live imaging of bacterial biofilms at single cell resolution using confocal microscopy. So here is an image of a mature Vibrio cholera biofilm. Okay? And what's very striking, what's sort of immediately striking about this picture is that is the dome-like shape. These biofilms grow to exhibit this very three-dimensional dome-like geometry that, for instance, a bacterial colony on agar would not develop, at least within the same time scale. And we now understand that this process is driven by cell verticalization. So basically, the cells within the biofilm will initially develop as a horizontal monolayer, but the accumulation of pressure within the interior of the biofilm will cause the cells in the middle to stand up, to verticalize in this way, and project their offspring, to proliferate into the third dimension, thus forming this, this dome-like shape. So to go into a little bit more detail on how this works, initially the cells are all horizontal, and they're growing and dividing, and so they're growing sort of into each other, and so they're pushing each other, and so they're exerting repulsive forces on each other, but they're also producing matrix, and the matrix is sticky. And so there is cell-substrate friction, which limits the outward motion of the peripheral cells. And that leads to the accumulation of pressure, to compression of the innermost cells. And once this compression reaches a certain critical threshold, these cells in the middle will verticalize. They will buckle out of the plane and verticalize, as shown here. And subsequently, there may be a second mechanical instability, this pinch-off instability, where further compression causes this vertical cell to be pinched off, ejected from the surface. But the energy barrier to this mechanical instability is actually quite high in real biofilms. So what we end up seeing in aggregate in an actual biofilm is this stably verticalized core of cells in the interior. So this is all fine and good. But all this work so far has focused exclusively on the substrate-bound morphology of biofilms with only strong cell-substrate adhesion and no cell-cell adhesion, which is also prevalent in biofilms. So we understand how cell-substrate adhesion plays a critical role in sort of dictating this morphology, this cleanly verticalized dome, But it's also natural to ask the question of what happens when cells can also adhere to each other and not just to the surface. And as it turns out, if we include cell-cell adhesion, if we allow the cells to also adhere to each other, biofilms can attain other morphologies. Namely, on the left, when we have just strong cell-cell adhesion alone, okay, then the biofilm becomes a floating ball of cells, which is somewhat intuitive. On the other hand, if we have both strong cell-cell and cell-substrate adhesion, we get the same similar sort of dome-like shape, but the cells along the substrate are less orientationally ordered. Okay, we don't have this clean verticalization that we see without cell-cell adhesion. So the questions that we have been focusing on in this work is, can we build a theoretical model that explains these effects? Can we fill in the rest of this phase diagram? Okay, we know what happens in these sort of extreme scenarios, but as we modulate the strengths of cell-cell and cell-substrate adhesion, can we see how the morphology of the biofilm changes? Okay. And can we then turn back to experiment and experimentally confirm the predictions from this model? So to do this, we turn to agent-based models. And I think the term agent-based model can mean different things to different people. But in our case, in these models, we model the cells as sphere cylinders that grow in length and divide and interact mechanically with each other and with the substrate. To be more specific, each cell is a sphere cylinder, as shown here, and is parameterized by its center, its orientation, which is a unit vector, its length, and its radius, which is fixed. So all cells have the same radius, and the cells are only growing in length over time. And so each cell grows in length according to this exponential growth law, which can be written in terms of the volume of the cell or the length of the cell. Okay. And upon reaching a critical length, the cell will divide into two parallel daughter cells. So far, very simple. So, but as the cells grow and divide, again, they are mechanically interacting with each other and with the substrate. And these forces give rise to a system of equations of motion for each cell. So I'm not going to go into all the details here, but to very briefly summarize, on the right-hand side, we have forces, generalized forces due to cell-substrate repulsion, cell-substrate adhesion, and cell-cell interactions here with neighboring cells. This is what I'm going to talk about in more detail in a minute. And then we have this additional term to account for this orientational vector norm constraint, because each orientation vector is a unit vector with a corresponding Lagrange multiplier. And on the left-hand side, we have additional forces owing to ambient viscosity within the bulk of the biofilm and cell-substrate friction for cells that are attached to the substrate. Okay. Now I'm going to go into much more detail on how we model these cell-cell interaction forces, which is sort of the new sort of aspect of this model. How do we model cell-cell repulsion and adhesion? So the first thing to keep in mind is that we should think of each cell as being encased in matrix, like this. So there is a soft coating of matrix around each cell. Okay, this is how we're going to represent the matrix in these simulations. And the cell wall itself is actually quite stiff, usually, but the matrix layer is much softer. So when two cells come into contact, this matrix layer is what is being deformed upon contact. Okay, so this is something that we should keep in mind throughout this entire talk. Now, without cell-cell adhesion, so in the baseline case where each cell-cell interaction is purely repulsive, a standard choice is a Hertzian contact. So this is a very widespread use assumption in these types of models. So the force between cell I and J, the repulsive force, is given by this formula here, which whose magnitude scales with the 3/2 power of the overlap distance, delta ij, between the undeformed profiles of the two cells at the contact point, like this. Okay. We can alternatively rewrite this force in terms of the contact radius, A, which is given here. But one key thing to keep in mind, and sort of a theme that will recur throughout this talk, is that this is an approximation, because what this formula is really the Hertzian force between these two spheres at the contact point. Okay. But to be fair, this is an approximation that is very widely used in the literature, okay, and it clearly has not gotten in the way of sort of recapitulating key experimental findings throughout the years. So this is a very widespread assumption. Now, building on top of this, if we were to build on top of this to incorporate cell-cell adhesion, a natural choice is the JKR model. It's the Johnson-Kendall-Roberts model, which is a very canonical model of adhesive contact. Okay. So again, what I'll do here is take the spheres at the point of contact between the two cells, and I'm going to I'm going to assume that the two spheres are in adhesive contact with each other rather than just purely Hertzian repulsive contact. And so the force between the two cells is given by this formula here, okay, where we have this positive repulsive term, which is a Hertzian term. It looks very Hertzian here. But there's also this counteracting adhesive term, which depends on this parameter gamma, which is the adhesion energy density that basically defines the strength of the adhesive contribution. And the key variable here is A, which is the contact radius. Okay. Now, but the contact radius in the JKR model follows a more complicated relationship with the overlap distance delta ij, as shown here. So in practice, for any two cells in the simulation, we know what delta ij is. We know what the overlap distance is. So we need to rearrange this equation and solve for A, which we can do, and then we can plug that back into this expression to get the force. Okay. So this is what we would do in practice in a simulation. So for a quick primer on the JKR model, I'm plotting the contact radius and the force as a function of the overlap distance. And I'm setting some of the parameters to physiologically relevant values. But I'm also, varying gamma, the cell adhesion energy density, to achieve particular equilibrium overlap distances. So for instance, if I set gamma to this value here, then I achieve an equilibrium overlap distance of 100 nanometers. The force is 0 at 100 nanometers. So this is how I'm going to tune, throughout this talk, the strength of cell-cell adhesion to achieve various equilibrium overlap distances. Okay. So this is what that is. And to very quickly summarize, the force is adhesive for small overlaps and repulsive for large overlaps. So when the force is negative, it's adhesive. When the force is positive, it's repulsive. Okay. The force predicts a discontinuous jump in the force at 0 overlap. So as soon as the two cells come into contact, there is a non-zero force, instantaneous non-zero force. Okay. And for anyone that's familiar with the JKR model, they'll know that the model actually also predicts hysteresis at negative overlaps for cells that had been previously contacting. Okay, if you pull them apart, there's also some residual adhesive force, but for the sake of simplicity, we ignore this throughout in this work. But again, one thing to keep in mind is that this is, again, an isotropic model, okay, in the sense that, again, I am really calculating the JKR forces between the two spheres at the contact point. So all of these cell-cell configurations are subject to the same JKR forces. And this is perhaps this isotropic assumption is perhaps a more reasonable assumption in the case of cell-cell repulsion with a Hertzian contact, because with a Hertzian contact, the deformation is assumed to be quite small. But with a JKR contact, as shown in this plot on the left here, the contact radius can be actually quite large. And the deformation is quite large sometimes. And so perhaps this is a bit less valid of an assumption to make in this context. And so to circumvent this, we also built a second model, a fully anisotropic JKR model. And the approach we take here is to transform this contact problem into an equivalent ellipsoidal surface contact problem, okay, where an ellipsoid is in adhesive contact with a surface with an elliptical contact region. Okay. And we solved this problem using a model developed, recently proposed by Giudici et al. In this paper. Which is actually an updated version of an earlier model by Johnson and Greenwood here. Now, this model is much more complicated. It's very elaborate. Okay? But just to briefly summarize, this model yields forces that also depend on the specific contact geometry. So you can imagine sort of fixing this blue cell here and achieving various contact geometries, a family of contact geometries, by sliding this green cell along, this blue cell. And what we see here is that the force obtained through this model depends not only on the overlap distance and on the equilibrium overlap distance, the cell-cell adhesion strength, but also on the specific contact geometry. So we have two models of cell-cell adhesion. We have this quasi-isotropic spherical model of cell-cell adhesion, which is based on the classical sort of spherical JKR model, and this second fully anisotropic model of cell-cell adhesion. So now we are ready to see some results. So here are some simulations I'm going to show you now. And I'm going to start with this baseline case where there's no cell-cell adhesion and just strong cell surface adhesion, cell substrate adhesion. So this is the case that has been well characterized in the literature already. And we see this dome shape that I've talked about already. Right? Now I'm showing the same simulation from below. Okay. So I'm looking at the bottom of the biofilm, and I'm coloring the cells by Z orientation. Right? And for all these simulations that I'm showing from below, all the cells that are further from the substrate than 0.4 microns are in light blue. So you can see some light blue cells behind, but all these cells in front are really the bottom layer of cells. Okay. And we can see, if I replay part of this video again, we can see that we have this verticalized core, and we really only have horizontal cells in white and vertical cells in purple and nothing in between. Right? And whenever a cell verticalizes, it verticalizes almost instantaneously. It's like it snaps into its vertical configuration. So this is the case with no cell-cell adhesion, only strong cell-substrate adhesion. Now what happens when we introduce cell-cell adhesion? So a weak amount of cell-cell adhesion. What we see is a similar morphology, but now we're starting to see the cells actually rotate. So now the cells are verticalizing more slowly with weak cell-cell adhesion. And if we strengthen the cell-cell adhesion, what we see is something different, which is that we see these light purple cells. We see these cells that are neither horizontal nor vertical, but actually are adopting sort of intermediate tilted configurations. So we see a greater diversity of Z orientations along the substrate, and this is exactly what I talked about previously that we know from experiment occurs. We see this greater degree of orientational disorder along the substrate, okay, with cell-cell adhesion. So to summarize, this is what we see, right? The y-axis is time, and each row in this heat map is a distribution of Z orientations at each time point. And so what we see here is with the onset of, initially we have this totally horizontal population, and with no cell-cell adhesion, we get this very clearly bimodal distribution of horizontal cells and vertical cells. But as we introduce and strengthen cell-cell adhesion, we get this more diffuse distribution of Z orientations along the substrate. Now I'm showing the same simulation, but with the cells colored differently. Now I'm showing the cells colored according to their minimum Z coordinate, which I'm showing schematically here. And what you'll also notice here is that we see these little patches along the substrate where there are no cells in contact with the surface. Okay. So these dark green cells, but also these light blue cells that are even further away from the substrate. In other words, with sufficiently strong cell-cell adhesion, we begin to see delamination from the substrate. Now, the next thing we can do is weaken cell-substrate adhesion while maintaining strong cell-cell adhesion. So what happens then? Now we begin to see proper delamination of the biofilm from the substrate. So we see this big void in the middle of the biofilm. Okay. If we weaken cell-substrate adhesion even further, then we will still get delamination. But we also see something in addition to that, which is that the biofilm starts to spread further, and that is because the cell-substrate adhesion is coupled to cell-substrate friction, right? And so because the cell-substrate friction has been lowered, the cells will start to spread further. Okay? And so the onset of verticalization, the onset of delamination is also delayed. And if we weaken cell-substrate adhesion enough, so 50 times its wild type value, or 1/50th its wild type value, then we just get a monolayer of cells. So to summarize, what we're seeing here is that if we have strong cell-cell adhesion, and if we weaken cell-substrate adhesion, okay, so each plot here is a similar plot to what I showed before, but here I'm showing the distributions of minimum z coordinates at each time point. And how to read these plots is that basically there are three subpopulations at play. There's this horizontal population, okay, that's attached to the substrate. There is this population here of cells that are tilted or vertical, but still attached to the substrate. And there's this population of cells that are suspended, that have been delaminated from the substrate. And what we see is that as we weaken cell-substrate adhesion more and more and more, we get more and more cells in this subpopulation of the biofilm. But we also delay the onset of verticalization and delamination, right? Because the biofilm can spread further because of weaker cell-substrate friction. And finally, one last simulation to show you here. What if we have no cell-substrate adhesion and only strong cell-cell adhesion? Then we see that the biofilm begins to behave as a liquid droplet. So all in all, we have this morphological phase diagram where we have this cleanly verticalized core over on the bottom right, and then as we increase cell-cell adhesion, we start to see a greater diversity of Z orientations along the substrate. As we weaken cell-substrate adhesion, we begin to see delamination. And then on the top left, when we have only cell-cell adhesion, we see a liquid droplet, basically, a biofilm that behaves as a liquid droplet. If we use the anisotropic model of cell-cell adhesion, we get basically the same results. So even though the model is quite different, we see that qualitatively speaking, we obtain the same morphological phase diagram. So the two models sort of agree in that regard, okay? And we are now actively working on understanding how these morphologies emerge. So we're working on sort of building toy mathematical models that sort of get at the mechanisms underlying these morphological transitions. But to give you sort of a schematic picture of why this is happening, let us revisit the verticalization instability that I talked about previously. If we consider the configuration immediately after the central cell buckles, okay, and if we pay attention to the configuration of this cell on the right, okay, this cell is subject to cell-substrate adhesion forces in yellow and cell-cell adhesion forces in orange, okay, and there is a competition between these forces. So if cell-cell adhesion is much stronger than if cell-substrate adhesion is much stronger than cell-cell adhesion, we're going to see verticalization because this cell on the right is not going to care about the cell in the middle, and the cell in the middle is free to verticalize of its own accord. Okay. On the other hand, if cell-cell adhesion is much stronger than cell-substrate adhesion, you can imagine that this cell in the middle is going to sort of tear the cell on the right off the substrate, like this. And anywhere in between, we might have this more intermediate tilted configuration, okay, where the cell on the right wants to stay attached to the substrate, but also wants to stay attached to the cell in the middle. Okay? So we can see, even from this very simple schematic, how the verticalization, tilting, and delamination arise from this competition between cell-cell and cell-substrate adhesion. One last point that I would like to make is that in this morphological phase diagram, I've already shown you we know from experiment that we do obtain this liquid droplet on the top left, and this orientational disorder along the substrate on the top right. But this delamination morphology is something that I have not talked about in any experimental capacity. But as it turns out, this is something that happens experimentally. So deep within the SI of this paper that was recently published from our lab, this is an image of a Vibrio cholera strain, a biofilm formed by a Vibrio cholera strain that has strong cell-cell adhesion, but intermediate levels of cell-substrate adhesion. And lo and behold, we do see this delaminated core in the middle of the biofilm. So this is something that we are looking to sort of characterize in much greater detail this strain and other related strains to see sort of how this delamination arises in real life. So to briefly summarize, cell-cell adhesion can very strongly influence biofilm morphology, as shown here. And using our agent-based model, we have been able to predict a morphological phase diagram, where we have, with strong cell-substrate adhesion, we have a cleanly verticalized core. Increasing cell-cell adhesion causes basal cells to adopt tilted configurations. Decreasing cell-substrate adhesion from there causes delamination from the substrate. And strong cell-cell adhesion alone yields a droplet-like morphology. And now we are working on sort of along both theoretical and experimental directions to sort of understand these morphological transitions in greater detail. Okay. So I think I've run out of time. I was going to talk very briefly about another direction, but basically just a one-sentence summary is that we can use the same techniques, we can use the same agent-based models to study phenotypically heterogeneous biofilms, pattern formation in phenotypically heterogeneous biofilms, and understand how different patterns arise in biofilms that have different phenotypic states. But this is maybe a discussion for another time, and I think I've run out of time now, so I'll stop there, and I'd be happy to take any questions.
All right, so applauses to Chris's very nice talk. I see one question now in the chat from Miranda Lynch. She says, wonderful work in the reduced cell-cell adhesion. How do the regions of adjacent vertical cells form? The upright forest of the entirely vertical cells or something else? So it isn't clear to her how a fully verticalized cell in the center would push up an adjacent non-vertical cell. And, how are these cells associating with each other in the core, basically about, how the verticalization happens.
So that's a very good question. So I think something that I did not necessarily show in the schematic, but something that does happen in the simulations is that I think it's a rather complicated process in the sense that if we look at this simulation with no cell-cell adhesion, then there are still horizontal cells in the middle here that are, so it's not a cleanly verticalized, it's not just a core of entirely verticalized cells and a subpopulation of peripheral horizontal cells, but we do have horizontal cells sort of in the center as well, and that I think is also sort of helping some cells in the that also acts to sort of exert pressure on horizontal. So here, for instance, we might have this cell verticalized upon division because of the growth-induced compression due to this cell. But I think that's a very interesting question. It's not something that we understand very fully, I think, even now. So there's been a lot of work on this subject, but I think that's a very interesting question. That's something that we should try to understand in greater detail.
Chris, I also have sort of a clarification question. Maybe you said it. You mentioned that you tried anisotropic cell adhesion and the results was pretty much the same. So how exactly do you define anisotropic cell-cell adhesion?
Yeah, that's a very good question. So so I can show you. So there's a ton of slides that I've skipped over here, but basically there is a pipeline that I introduced here, where basically I am going to take this, take any pair of contacting cells, and use the overlap distance and the angle between these two cells, and also the principal radii of curvature at the contact point. So there are 4 principal radii of curvature. And I am going to use those values to convert this contact problem into an equivalent surface contact problem according to this sort of standard procedure that was developed by, I believe, by Landau, Lifshitz, or Johnson, or someone, I can't remember. But there is a well-established procedure for performing this transformation. And then now we follow the prescription given by Giudici et al. To calculate this force here, the adhesion force here between this ellipsoid and the surface, which depends on the overlap distance as well, delta here, and the two principal radii of curvature at the contact point. And the formulas here are rather very complicated, but basically the first thing that you do is you calculate the aspect ratio of the contact area, and then you use that aspect ratio to calculate the force.
So it's an anisotropy with respect to geometry.
Yes. Yes.
Okay.
That's right.
Okay. Thank you.